and you will find them as separate recordings in the Kaltura course gallery. Okay, just to make sure, I'm asking again our friends on Zoom if they can see the first slide with the introduction to histology that tells me that I'm sharing the screen. Can you see it? Folks, you can still hear me well. Can someone? Yes, I see already. Thank you. So we begin with, I'm telling you a few words about the histology and the methods that histology uses to look at the organization of the tissue. And every time I'm the first slide, I will also point to the chapters from the Nether's Essential Histology and from Kera's Basic Histology that deal with the topic. There is, after the first slide, there is another slide that it usually focus your attention on the main things that we should retain, the take-home message for these lectures. So in this lecture, we will know what histology studies, we will know the techniques used to investigate tissue organization, and there are microscopy. It's the technique that we're using, and there are different types of microscopy. We will use mostly light microscopy, but I will quite often also show you images of better resolution, and we'll see what resolution means, of electron microscopy. We will look at the most used stains. The most important stains that histology uses is H&E, hematoxylin and eosin, and it highlights very well the cells, the intracellular organelles in cells, and it allows you to distinguish between the different cells and between the cells and the extracellular matrix. The tissues and the organs, everything in our body, it's actually made of cells, and the material that is in between cells, the extracellular matrix, that is always secreted by the cells. We will learn how to recognize a cell or a structure in a tissue section. And it's the best thing how to learn how to do that is by showing you perfect examples that I may take them myself from the virtual microscope or from the textbooks. Sometimes even that is not sufficient, and we need to rely on some drawings. But after a time, you will get used to recognize the boundaries of the cell, even if they cannot be clearly identified, but you sort of guess that those have to be the boundaries of the cell. You will definitely see the nuclei, and you will see the contacts between cells, and this is sometimes a big problem for some students who never looked at a micrograph in the past. But you will very fast learn how to do that. The histology has several branches, histochemistry and cytochemistry, and this is, it's actually the histochemistry and the cytochemistry, they are the same thing, but they focus either on tissues, histo means tissue, and on cells. And they do more than, they want to achieve more than just to stain the cells and the extracellular matrix in a sort of indiscriminate way without focusing on particular proteins or lipids. And that is usually done with antibodies that are raised to specific molecules. And So they identify the chemistry of the components that are present in the cells. So sometimes if there is a text slide, I will just read from it to make sure that we get fast. So histology is the study of the tissues of the body and how these tissues are arranged to constitute organs. It is a visual science which aims to assess the functional states of cells and tissues of the body. The images that you will see here are micrographs, which is photographs obtained through a microscope. And in the past, those photographs used to be real photographs taken on a photographic film. However, these days, all these photographs are digital micrographs that we can watch on the screen. So the microscope is essentially a magnifying lens. Histology, the name histology implies that it focuses on tissues. Histo means tissue in Greek, I think. But so the focus is on tissues such as the epithelial tissue, but the tissues are made of cells and the intercellular matrix, like the acinar cells, for example. If we focus on the pancreas, the tissues are part of organs, the organs are part of systems, and the pancreas is part of the digestive system. In part, there is the pancreas has also structures that produce hormones, and so the pancreas is also an endocrine organ that is part of the endocrine system. And then all these systems, they are putting together our organism, and they obviously they interact with each other. So the goal of histology is to take a piece of tissue and look how it is organized. You pick that piece of tissue through a biopsy. Or if you do use animals for scientific research, you may take a piece of tissue from the animal. And that piece of tissue needs to be then prepared to be able to be viewed with a microscope. And that essentially means that the tissue needs to be hardened, embedded in something that is hard, so that it is cut in very thin sections. Because the microscope, in order to provide the information about the individual cells, needs to look at a very thin section from the tissue. And this is an important slide that illustrates the steps that one needs to do from collecting the piece of tissue till cutting the embedded tissue with an instrument that is called microtome. The microtome has essentially a blade and cuts very, very thin sections of the embedded tissues. And when I say thin, that means that the sections can be 10 microns, 25 micrometers, 50 micrometers, sometimes even 100 micrometers, but not thicker than that, because one cannot then separate, unless one uses confocal microscope, the individual cells. But let's see how we need to process this piece of tissue that we take from a research animal. When the tissue is cut, cells are lysed, and the cells contain many enzymes, many proteases that start chewing up the proteins that are present in this piece of tissue. And that needs to be stopped. And one does that by fixing the tissue. And the fixation actually aims at immortalizing the structures as they are when they have been picked up from the animal. the main fixatives that are used in histology are aldehydes and formalin. It's a solution of 10% formaldehyde. That is a good fixative in most cases, and it is an aldehyde, it polymerizes, so you have several aldehyde groups that are kept together in a longer polymer, and the aldehyde groups will bind to the amino groups in proteins, and they will cross-link the proteins and keep them in place. Now, if one would like to look at the composition of these fixed pieces of tissue and shine light on them, the light will be sort of dispersed in all directions, and it will make the piece of tissue translucent, look milky. Not the idea would be to make it transparent so that the light passes through the tissue and we can identify the various things that are present there. This translucency, it's given primarily by the lipids that are present in the tissue. And so the next step has to take into consideration that it is good for the lipids to be removed from the piece of tissue, and also it takes into the consideration the fact that one needs to permeate, to infiltrate the tissue with something that can harden and generate a hard tissue block. And that's something, it's usually a wax, and the wax that's used in histology, but most of the time it's paraffin. Paraffin, it's solid at room temperature, but around 50, 60 degrees Celsius, it melts and it can enter and infiltrate the tissue. So the first thing, however, is for the fixed tissue, it's to dehydrate it. And the dehydration, it's done with ethanol. And the piece of tissue is passed through solutions that contain increasing concentrations of ethanol. Ethanol, it's an alcohol, and it removes the lipids, and it replaces the water. But the next step would be to infiltrate the tissue with molten paraffin. That cannot be done, however, when the tissue is infiltrated with ethanol, because the molten paraffin, it's not intermixable with ethanol. So what one usually does, it's replacing the ethanol with another solvent that is usually xylene, and xylene, it's intermixing with the molten paraffin. And this step, so this is the dehydration step. The next step, the infiltration with xylene, it's called clearing step. Why? Because the xylene, it has a high refractile index, it penetrates through the piece of tissue, and it makes the tissue now transparent to light. Light passes easily through the piece of tissue. So this is the clearing step. The next thing is to replace the xylene with the molten paraffin. And this is the infiltration step, which is different from the embedding step. The embedding step follows the infiltration step, and it is just the cooling down of the embedded tissue, and the paraffin will now harden. One can trim the tissue block and generate now the final tissue block that can be put into the microtome, and one can cut paraffin sections. Of course, the stains that are used in histology, they are mostly water-based stains. They are made in various buffers, and they will not enter the paraffin sections. So one needs to go and process the paraffin embedded tissue in a reversed way so that one gets in the end a tissue that is present in a buffer, watery buffer that can be subjected to staining. And of course, one removes the paraffin with xylene, they replaces the xylene with ethanol, and then goes now to decreasing concentrations of ethanol to finally reach not the solution that contains the fixative, but a buffer that and the sections are usually collected on a glass slide, and they are processed for staining on the glass slide. But what so we have now the specimen, it's called the specimen that is on the glass slide, and we need now to look at it through the microscope. This is the microscope, the basic, it's a drawing of a microscope, but the basic features are this. So this is the stage. One puts the glass slide here, the specimen is here just in the path of light. The light it's usually emitted from a tungsten halogen lamp. It is condensed and focused on the specimen. It passes through the specimen. Light is now dispersed after interacting with the various structures that are present in the specimen, and then the light is then reassembled into the image of the tissue by this most important part of the microscope, which is the objective. And the image can then be magnified more through the ocular, and it is then focused on the retina if we look by naked eye through the microscope, or on a screen of a digital camera, and one collects the image as a digital file. So the main features of a microscope are the magnification. The magnification is primarily given by the objective. And there are objectives that we have in the lab, a 10x objective, and we go up to 100x objectives. We have 10, 20, 40, 60, and 100x objectives. But the magnification is not really the most important characteristic of the objective. What is even more important is the resolution that the object offers. And you can imagine that a high resolution will allow to distinguish of two spots in your specimens that are separated by the smallest possible distance. Beyond that, if the two spots are closer than that maximum resolution that a microscope can achieve, they cannot be seen as a single spot. So the resolution is the smallest distance between two structures at which they can be seen as separate objects. The resolution, the smaller the resolution, the better it is. The maximum resolution that is obtainable with a light microscope is 0.2 microns, and this is actually set because of the characteristics of the visible light, and I'm referring here to the wavelengths of frequency. And the best resolution obtainable with visible light is 0.2 microns. The 0.2 microns are the smallest distance that two structures can be apart from each other so that they can be seen as two different structures. The small resolution it's also the highest resolution, but you need to keep in mind that there is something that is the resolving power. The resolving power is inversely proportional to the resolution. But what you should keep in mind is that the resolution, it's the maximum resolution that can be obtained in a microscope. It's set by the characteristics of the light. You know that the light has a dual nature. It's a particle, the photons, and they have an associated wavelength. The smaller the wavelength, the higher the resolution is. That is the smaller is the distance that two structures can be still seen as separate structures. The human eye, it's really not a very, has not a very good resolution. It's a wonderful resolution for what we usually need to do. And we don't look with a microscope, we don't look at the intimate structures of the objects. But the human eye has a resolution of 0.2 millimeters. So if two particles are separated by less than 2 millimeters, you won't see them as separate particles with the naked eye. However, the transmission electron microscope and even the scanning electron microscope, they can achieve resolutions that are, the transmission electron microscope has a resolution of 0.2 nanometers. It's about 1,000 times a better resolution than the one that is achieved with light. And this is possible because the transmission electron microscope uses waves, but they are not light waves. They are electron waves. Each electron has an associated so-called de Broglie wavelength. I think I have it somewhere later on written. I'll tell it when we get to that point. But because the wavelengths associated with an electron, it's about 1,000 times smaller than the wavelengths of light, the electron microscope can achieve a resolution that's 1,000 times higher than that of a light microscope. Now, if you just put under the microscope a specimen that was embedded in paraffin, even if you remove the paraffin, you put it under the microscope, you won't see much, because now the light will pass, the lipids have been extracted, and the light is not absorbed in particular by any of these structures. And usually the image, a good image, is obtained if one can generate a contrast between the various structures that are present in your specimen. And the bright field microscopy cannot generate such a contrast. There are some cells. These are neural crest cells. We'll see, I think that 2 weeks from today, what neural crest cells are. They are the precursors of the cells that form the peripheral nervous system. But there are some tricks that can be done with the microscope so that one can look at cells in unstained, non-stained specimen. And those tricks rely on the properties of light. And this is the phase contrast micrograph. The light waves have several features that characterize that. They have a certain wavelength that it's sort of inversely proportional to the frequency. They they have an amplitude, and the amplitude is really important, that needs to be changed so that we can create contrast. They have a plane of oscillation, and they also have a characteristic that actually comes from comparing two light waves, for example, that travel through the specimen, and that is the phase. The two waves can achieve the maximum amplitude and the lowest amplitude at the same time or at different times. And one says that uh, the two light waves oscillate in phase or out of phase. And there are tricks that you can do with the microscope so that you can pass the light waves through a specimen, and they will be, the phase will be shifted in some of them and not in others, that according to what they pass through. And that difference in phase, the shift in phase, can be converted actually in difference in amplitude. And in this way you obtain the phase contrast image that allows you to see the cells, the nuclei. The light the light waves can interact with each other and may interfere with each other. And this light interference is used in another microscope setting, in the differential interference contrast microscopy, that also allows you to distinguish the cells. It's not a difference as much in the contrast between the various things in the specimen, but you get sort of a three-dimensional feeling of the images, of the DIC images, differential interference contrast images. However, to really be able to look at the structures that are present in a specimen, one needs to use special stains. And I mentioned already that the H&E stain, the hematoxylin and eosin stain, is the most frequently used to stain a tissue section. And there are two stains present in the H&E stain. One is the hematoxylin that has a blue color, violet color. It is said that it is a basic dye or cationic dye, and it binds basophilic, acidic, anionic, negatively charged groups that are present in DNA, in RNA, in the nucleic acids, that they contain those many phosphate groups, and also in phosphorylated proteins. And in general, the molecules and categories of molecules, they differ in their charges. And to remember for the exam, what you should remember about hematoxylin is that it binds negatively charged groups. Eosin, by contrast, binds positively charged groups. And there are positive charges in proteins. The lysine and arginine are positively charged amino acids, and proteins are in general more positively charged. And you'll soon see that collagen is one such protein that contains many positively charged groups that stain very well with eosin. Once the eosin is stained, it is an acidic dye, an anionic dye. It binds acidophilic, basic, cationic, positively charged groups. And here you have an example of a specimen from the small intestine. What you see here, so this would be the lumen, it's a cross section, and this is one side, this is the other side of the intestine, sort of facing each other. And what you really see here are one single layer of cells. And these cells are epithelial cells. They are the enterocytes. They are these are columnar cells. They have a so-called brush border formed of microvilli. But what you can see here is that their cytoplasm that contains proteins is pink, and that is because the proteins are eosinophilic. What is basophilic? Basophilic are the nuclei and the nucleolus. That is a site inside the nucleus. It's shown actually, it's even darker. There are some cells that do not stain with hematoxylin and eosin. And why does that happen? Because they are neutral. And these are the goblet cells, and the goblet cells secrete a mucus, secrete carbohydrate, neutral carbohydrates, mostly neutral carbohydrates. So coming back to the characterization of the structures that are stained with H and E, I note that the suffix philic in basophilic or eosinophilic is used to describe the properties of the structures being stained and not of the stain itself. The nucleus is basophilic and the cytoplasm is eosinophilic or acidophilic. Now coming back to those cells that do not stain with the H and E. They contain, however, carbohydrates, and the carbohydrates, the sugars, can be stained with the PAS stain, periodic acid Schiff, and it has this purple magenta stain. The periodic acid oxidizes the carbohydrates, and the Schiff reagents binds then the oxidized sugars. And see, this is now the staining of the goblet cells. There are also stains for lipids, and the stains for lipids that are mostly used are the oil red O. I don't have a slide for it here, but it should be red. It's Sudan black, which is also not shown here, and osmium tetroxide. Osmium tetroxide, it's also a fixative and a stain for electron microscopy. However, it is also a stain for lipids. And here you see that it stains large cells, the so-called unilocular adipocytes that are filled with a large droplet of lipids. This is a fascicle of myelinated axons, which forms a nerve actually, and the stain stains the myelin sheets that surrounds the myelinated axons contains a lot of lipid, and that binds osmium tetroxide. Osmium tetroxide binds the unsaturated fatty acids in the lipid. But if you would take a paraffin embedded section and you would stain it for lipids, you would not see anything. Why? You know why? Yes.
Because it takes away the lipids.
Exactly. The dehydration step has eliminated the lipids. And you will see many examples where the cells appear empty, not stained at all. However, if one embeds the tissue not in paraffin and avoids the dehydration step, let's say they are embedded in a plastic resin, as this specimen is here, or if one generates cryosections, then the lipids are preserved and can be stained. For cryosections or frozen sections, the tissue is fixed and immediately snap frozen sometimes even in liquid nitrogen. And the frozen tissue block can be cut in a special microtome that is called cryostat. And one can achieve very, very thin sections, cryosections. When I used to do such sections myself, I could cut even 2 microns cryosections. Now there are other stains I'm briefly mentioning the Nissl stain. This is used primarily in the study of the nervous system because it stains very well the large neurons. It is a blue stain, and it is actually a basic stain. It binds to negatively charged groups that are present, however, in the ribosomal ribonucleic acid, ribosomal RNA, which is part of the ribosomes. The ribosomes are part of the biosynthetic machinery for proteins. They can occur as clusters of ribosomes or polysomes in the cytoplasm, and there they translate the RNA into cytoplasmic proteins, or they may be associated with the rough endoplasmic reticulum, and there they translate the RNA into either transmembrane proteins or secretory proteins. So what you see here are large neurons stained with the Nissl stain. I mentioned already the virtual microscopy, the fact that you can look at a specimen and zoom in and out. It's because each of those specimens that they are actually electronic files. They are hundreds, maybe thousands of images that have been produced from the entire specimen and then reconstituted by a digitally to one single image. And if we have time, we'll go live into the virtual microscope and I'll show you how to zoom in that out. A few words about the immunohistochemistry and immunocytochemistry. I mentioned already that here one uses antibodies directed to specific antigens, specific molecules, to visualize those molecules in your specimen. And if you visualize, if you label the antibodies, if you covalently tag them with a fluorescent dye, you can view the image in fluorescence microscopy. And this is an example that I put together. This is a long axon of a neuronal cell that will grow in the lab, a CAD cell. This is the growth hormone, and we know that this neurite, the axons, it's actually packed with microtubules. And here the microtubules sort of split throughout the growth hormone. The microtubules are polymers of tubulin. So we used an antibody that detects, binds to tubulin, and that antibody is tagged with a green fluorescent tag. When they bind, we incubate the cells or the tissue with the antibodies and wash away the unbound antibodies. And when we look through the fluorescent microscope, this is what is seen. So the axon is heavy stained throughout its length because it's packed with the microtubules, but here you can see individual. I don't think that these are actually individual microtubules, but they are bundles of microtubules, and they run in all directions in the growth hormone. This is another image that you will use certainly at the TBL1 quiz, where I used an antibody to a protein, a molecular motor, a kinesin motor that it's KIR3A. I knew that it is present in the retina, but I did not know where it is localized. And I used immunohistochemistry with this antibody and detected it with a fluorescently tagged secondary antibody. And I found it localized in this region, sort of in the middle of this region, and also in the middle of this region. And there is nothing that you can really identify there, but you will see that you will know by the time of the TBL1 that these are the various layers in the retina. And this is the pigmented retinal layer, the layer of rods and cones, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, and nerve fiber layer. And you clearly see that this staining is present in the outer plexiform layer, and this staining is present in the inner plexiform layer. Please keep in mind this image from the TBL one, please. Now, very few words about the electron microscopy. I mentioned already that the resolution of the electron microscope is about 1,000 times better than the resolution of a light microscope. And I'm saying that the resolution of a transmission electron microscope. And this is because the electron associated wavelengths, the Broglie wavelengths, the electron associated wave has a wavelength that is about 1,000 times smaller than the wavelengths of the light of the that is used in the light microscope. This shows you two types of microscopes: the transmission electron microscope at left, the scanning electron microscope at the right. But let me focus first on the transmission electron microscope. Now, the electrons are actually the ones that need to be produced, and they are emitted from a cathode. They are charged particles, negatively charged particles. They are accelerated against an anode, and then they are focused by passing through something that is equivalent to a condenser lens. But instead of a lens, one uses a magnetic field that is generated here, and the magnetic field it's generated and focuses the electron beam on the specimen. Then there is the electrons are dispersed. The wavelengths of the electrons are dispersed and then refocused on an objective. These are actually coils, you know, that the coils generate electric current passes through them, and they generate a magnetic field, and that magnetic field leads to the focusing of the electrons. And the electrons then hit a screen that on an electron detector, which is usually a charged coupled device. And one records the image on the screen, usually. Of course, it can be also recorded on a film. But here you see the red blood cells, the discoidal red blood cells that are seen here. A variant of the electron microscope uses the same electron beam that is focused to these coils. It passes, however, not to the specimen. That the electron beam hits the surface of the specimen that is covered with a film of metal, usually gold, and the electrons are then reflected actually in various parts, and the reflected electrons reconstitute the image that appears sort of a three-dimensional image. So the scanning electron microscope, however, does not have such a good resolution as the transmission electron microscope. This is now a comparison, direct comparison between the light microscope and electron microscope. The objects, the specimen is placed here. The This is the light source, this is the electron source, this is the coil that serves as a lens, similar to a lens for the electron beam. Another coil, this is the objective coil. And now you can see the difference between what you can see in a light microscope. These are chondrocytes, the major cells, or in cartilage. You see the boundaries of the cell, you see the nuclei, but no detail inside the cell. However, this is an transmission electron micrograph. You see the nucleus with darker and the lighter parts, the heterochromatin and the eurochromatin, various membrane-bounded structures. So this would be the cytoplasm. The darker areas of the image are those that fewer electrons were transmitted through. The lighter areas of the image are those that more electrons were transmitted through. And I am telling you that the fixative, in this case, it has to be much stronger than the fixative used in light microscopy. And it contains the osmium tetroxide, which stains the lipids in light microscopy, and is a heavy metal compound. And other heavy metals like uranyl acetate and lead citrate are used as stains in transmission electron microscope. So remember for the exam that the fixative for the transmission electron microscope contains heavy metals as fixative, and they are also stains. I explained you the difference between the transmission electron micrograph and the scanning electron micrograph. Remember that only the transmission electron micrograph uses thin sections. The scanning electron micrograph does not. The electrons hit the metallized surface of the specimen and are reflected and reconstitute the image. And this is a comparison of a transmission electron micrograph of a chondrocyte, and this is a scanning electron micrograph. And you see sort of the three-dimensional structure of the organelles, of the structures that are present inside the cells. And you may wonder, how is it possible if you take your specimen and metallize it, how is it possible for the electron beam to enter into the cells? This is not how it is done. One usually freezes the specimen and then fractures it. And when it fractures, the fracture line may break through the cell and expose the inside of the cell that can be then metallized with a thin layer of gold usually. Now, we'll take a break. After I finish this lecture, I still have to go through the cell part. What are the cells and the main features of the cell? Then we'll take a break and continue with the epithelial tissue lecture. And here we will look and understand the principle omnis cellula et cellula. All cells come from cells, which is something that humans really realized quite late in the history of the society. And it was the German histologist and pathologist, also Rudolf Virchow, that came to the conclusion that at that time it was already realized that all the organisms are made of cells, but it was not known how those cells are produced. And Virchow was the one that realized that there is no cell produced de novo. Every cell originates from a precursor cell that divides. And the first cells emerged on Earth have it written here, 3.8 billion years ago. And that is only 700 million years after the Earth was formed. These are things that I'm sure that many of you thought of these things, that about 13.7 billion years, everything that we is around us, our entire universe, was concentrated in what's called today a singularity, something that is almost dimensionless. All the matter was concentrated there. There was no space there, there was no time. And suddenly the Big Bang occurred and generated the entire universe. And then it took quite a long time until the planets and the Earth was formed. And the Earth was formed about 4.5 billion years ago, but only after that, it took only 750 million years for the first cells to appear. And once they appeared, that was when all cells that are present today have been produced de novo. Since then, the cells that were generated were produced through cell division from those cells that appeared at that time. Isn't that amazing? It's fascinating. So we will then look at the organization of the cells and relate the structure of the intracellular components to their function. And we will understand how cells become organized into tissues. And I will sort of explain you many things about cells as we go along through this course. But today I will briefly tell you just a few things. So the cells are the organism. Our organism, it's made of 4 basic tissues. The epithelial tissue, the connective tissue, the muscle tissue, and the nerve tissue. And all these tissues consist of cells and the extracellular matrix. I told you already that Rudolf Virchow came with the saying that omnis cellula e cellula, and that was based on what Schwann and Schleiden had found, that all living organisms are composed of similar units of organization called cells. And what Rudolf Virchow also realized is that the diseases originate in cells, in the cells themselves, not in tissues. And this is why he is considered the father of pathology. So the cell is the fundamental structure and functional unit of all living organisms. The eukaryotic cell consists of the protoplasm that is surrounded by the plasma membrane. The plasma membrane is a bilayer of phospholipids, and everything that's inside that enclosure forms the protoplasm. So inside the cell, there is the nucleus. The nucleus is surrounded by the cytoplasm. The cytoplasm contains the organelles and also some structures that are not bounded by membrane that are called inclusions. In the nucleus is bounded by the nuclear envelope and contains inside the nucleoplasm that consists of chromatin, the DNA, the nucleolus, and the matrix. Now, this is a two-dimensional drawing of a cell. The nucleus is this one. It's surrounded by the rough endoplasmic, smooth endoplasmic reticulum and rough endoplasmic reticulum. You have here the organelles, you have the cytoskeletal elements, and the plasma membrane or plasma lemma surrounds the cell. However, you have to imagine the cells as three-dimensional structures, and you have to get used to look at the three-dimensional structure based on the two-dimensional images that we look at in histology. Now let's take a look at the individual components of the cells, and we begin first with the plasma membrane that surrounds actually the what's inside the cell. The plasma membrane, I mentioned already, it's formed of two layers of phospholipids, and the phospholipid looks like this. It has a hydrophilic head that contains a negatively charged phosphate groups, and it has two tails, two hydrophobic tails, which are the fatty acids. And the way they are organized, these phospholipids, is with the heads towards either the interior of the cell or the other leaflet. The heads are oriented to the extracellular side. The fatty acids are inside, in between the heads of these two phospholipid layers. And in transmission electron micrographs stayed with osmium tetroxide. The osmium tetroxide also binds to the phosphorylated heads of the phospholipids and generates this structure. You see two dark lines that are less stained in the middle, and this is what's called the trilaminar unit membrane. This is how the plasma membrane of the plasma lemma that surrounds the cell, and all membranes that surround the organelles inside the cell have the same structure, a phospholipid bilayer, and appear the same in transmission electron microscopy. Here are cross sections actin filaments. But the current model of the organization of the plasma membrane is the fluid mosaic model of Zinger and Nicholson. When I went to school, and that I graduated from the Faculty of Physics, and in the last 2 years I specialized in biophysics. Then I was told that the model is that one of Dawson-Danieli, which still envisioned this phospholipid bilayer, but they thought that the proteins are actually attached to the bilayer. The fluid mosaic model of Zinger and Nicholson envisions the proteins either as spanning the bilayer, and these are the integral membrane proteins, or being attached on either side to the phosphorylated heads. And the integral proteins, they are contrasted with the peripheral proteins. And why is the model called the fluid mosaic model? Because these proteins, they are envisioned as diffusing in the plane of the membrane. So they are not static unless they are really fixed in place by the interaction with cytoskeletal elements or extracellular matrix proteins. But otherwise, they float in this lipid bilayer. Now let's take a look at the nucleus. This is a light micrograph. This is an electron micrograph. In light microscopy, you see the boundaries of the nucleus and some dark, very basophilic spots, and they correspond probably to the heterochromatin. But the real organization of the chromatin inside the nucleus is shown very nicely in a transmission electron micrograph. So the dark spots are the compact chromatin, the highly compacted chromatin that is not transcribed into RNA or replicated as it happens during the S phase of the cell cycle, and the bright parts that is unwound chromatin that is undergoing transcription into RNA, or it may be replicated to duplicate the genome before the mitosis. There is also this dark structure, large dark structure that is the nucleolus, and the nucleolus is the site of putting together the subunits of the ribosomes. I mentioned the ribosomes, and the ribosomes are structures that are formed from proteins and ribosomal RNA. And the ribosomal RNA is a structural component. And I insist on that because the ribosomal RNA actually binds the Nissl stain that I talked about. So the nucleus plays roles in replication that leads to genome duplication, transcription during gene expression, and ribosome production in the nucleolus. It's showing you a cell that has a lot of the chromatin compacted as heterochromatin. This is not undergoing robustly protein synthesis. If neurons often are engaged in robust protein synthesis, and most of the chromatin it's decompacted and becomes euchromatin. Here, this is everything except for the nucleus. It stains slightly. Now the membrane that surrounds the nucleus is actually a double membrane, and you see it in transmission electron microscopy, one part and the other part, the double membrane. And what you see here, this disruption, the absence of connection of these membranes with these membranes is because there are nuclear pores. In a scanning electron micrograph, the nuclear pores appear like this. They are really holes in the nuclear envelope. It's called the nuclear envelope. And this is necessary because there is a lot of traffic out of the nucleus into the cytoplasm and from the cytoplasm into the nucleus. And those large subunits of the ribosomes, they are produced inside the nucleus and have to exit into the cytoplasm and form the polyribosomes and attach to the endoplasmic reticulum to produce the rough endoplasmic reticulum. Now you know that the nucleus disappears actually during mitosis when the mitotic spindle forms the centrosomes. They duplicate and they migrate at the opposite poles of the nucleus that is disintegrated. By that time, the chromosomes are assembled and cell division occurs. This is the M phase, and after the cells have separated their genetic material, they undergo cytokinesis and become to separate cells. Let's now take a look at the organelles that are present in the cells, and I usually look first at the organelles that where the proteins are synthesized, and that occurs in the rough endoplasmic reticulum, and I'm referring here to the transmembrane proteins and secretory proteins. This would be the nuclear envelope, and the endoplasmic reticulum actually continues the nuclear envelope, and the endoplasmic reticulum is formed of membrane tubes and membrane cisternae that are connected with each other. The proteins are synthesized in the rough endoplasmic reticulum. They are then recruited into transport vesicles and transported to the Golgi apparatus, where they are modified by glycosylation and sulfation, and then they are recruited in transport vesicles in the cis part of the Golgi apparatus, and these transport vesicles carry the secretory product to the periphery, to the plasma membrane, where these vesicles fuse actually with the plasma membrane, and some of them carry the hydrolases, various enzymes that cleave the proteins, the carbohydrates, the lipids, and carry them to the lysosome. So this is the biosynthetic secretory pathway. There is the endocytic pathway that takes material from the extracellular space, internalizes in endocytic vesicles, and transports them to the lysosome. There is also a retrieval pathway, but I don't insist on them. Now take a look at the endoplasmic reticulum. The smooth endoplasmic reticulum lacks the ribosomes attached to its membranes. The rough endoplasmic reticulum contains the ribosomes attached to its membranes. This is a stained section where the endoplasmic reticulum it's stained in red. So both types of endoplasmic reticulum have a biosynthetic function. The smooth endoplasmic reticulum is the site of lipid synthesis and carbohydrate metabolism. The rough endoplasmic reticulum synthesizes proteins for secretion, incorporation into the plasma membrane, and as enzymes within the lysosomes, as I've shown you there. So they have also a storage function, but I stress on the detoxification function that is a feature of only of the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum is the site of lipids biosynthesis. It's the site of detoxification of various toxins, and it is also a site for calcium to be stored. This is a transmission electron micrograph. These are the smooth endoplasmic reticulum cisternae. And this is a drawing that shows the smooth endoplasmic reticulum on the outer part and the rough endoplasmic reticulum in the inner part. The rough endoplasmic reticulum in transmission electron microscopy appears also as tubules and cisternae, but it has attached those dots to it, which are the ribosomes. The ribosomal subunit are actually produced in the nucleolus. This is a Nissl stain, this is the nucleolus. And the Nissl stain binds to the ribosomal RNA that is present in the ribosomes. So the Nissl stain stains the polyribosomes in the cytoplasm that are clusters of ribosomes and the rough endoplasmic reticulum. Then the proteins are transported through secretory vesicles, not secretory vesicles, through transport vesicles from the endoplasmic reticulum, rough endoplasmic reticulum, to the Golgi apparatus. The Golgi apparatus is formed of stacked cisternae, stacked saculae, and they have, this is how they appear in transmission electron microscope. The Golgi has a cis site, and at the cis site arrive the transport vesicles from the endoplasmic reticulum. They pass through the Golgi apparatus and reach the trans site, where the proteins are packed into transport vesicles in a region that is called trans-Golgi network. So here it's where those proteins are assembled into these transport vesicles. I mentioned that the addition of carbohydrates, which is a post-translational modification of the proteins, occurs in the Golgi apparatus, and also the addition of sulfate group phosphorylations. Now, this is a transmission electron micrograph showing the transport vesicles that emerge. They come from the trans-Golgi network and reach the either the plasma membrane or the lysosomes. And the eosinophilic staining that is seen here in the pancreas, in this pancreatic acini, comes from the staining of these secretory vesicles that are called zymogen granules because they carry the zymogen, the precursors of the active digestive enzymes that are delivered to the duodenum. Now, the secretory vesicles reach the plasma membranes and they undergo the fusion. The membrane of the vesicle fuses and becomes continuous with the plasma membrane, and the secretory product is released into the extracellular space. And this is an example of such a fusion is at the synaptic terminal. Here you see the synaptic vesicles accumulating at the synaptic terminal, and they will release the neurotransmitter after they fuse with the plasma membrane of the axon at the synaptic terminal. However, if you see such vesicles that are attached to the plasma membrane, they may also be endocytic vesicles. Now the endocytic vesicle will form by invagination of the plasma membrane, and those vesicles will detach from the plasma membrane and become endocytic vesicles that take material from the extracellular space inside the cell. And this is the endocytic degradative pathway. It's shown here in green. And this pathway, it mostly starts at the cell periphery and internalizes material from the extracellular medium, or it may start inside the cell. And the destination of all the material that is taken up into such vesicles along the degradative pathway and in the lysosome. The lysosome is an organelle, the site where the proteins, the lipids, the carbohydrates are hydrolyzed to building blocks, to simple molecules. There are proteases, there are glycosidases, lipases, phosphatases, sulfatases, nucleases, and those are they reach the lysosome by transport from the Golgi apparatus to the lysosome. See, these are the primary lysosomes. Now, the endocytic pathway may take up large particles like even bacteria, and that process is called phagocytosis, and the phagocytosis occurs by taking up those phagosomes into phagocytic vacuoles. The phagocytic vacuoles then fuse with the lysosomes, and the bacteria are degraded to simple amino acids, simple sugars, lipids. Then the cells take up material from the extracellular space through small vesicles, small endocytic vesicles, through the process of pinocytosis. And in this way, they take from the extracellular medium the material that they will then transport to the lysosome and cleave hydrolyze the proteins, the sugars, and everything they took up from the extracellular space to use it to synthesize their own new proteins and carbohydrates and lipids. So the lysosomes cannot really be seen very well by light microscopy unless they are stained in a special way. However, here you can, all these blue structures, they appear as basophilic structures, they are the lysosome. These are these are fluorescently labeled lysosomes because they contain fluorescent particles that were taken up through phagocytosis and delivered to the lysosome. Now the power plants of the cell are the mitochondria. The mitochondria are the organelles that produce most of the ATP that is present in cells. So they are the energy factories in the cells, and they have these elongated structures. They are quite large structures. In transmission electron microscopy, one can see that they have a double membrane, and they have projections. The inner membrane projects into the inside of the mitochondrion with these crystals. And the mitochondria, this is not a very good image, but the mitochondria stay highly eosinophilic in light microscopy. This is a scanning electron micrograph showing the crystal that penetrate towards the interior of the mitochondrion. Now, a few words about cellular inclusions, which are also present in the cytoplasm of the cells, but they are not real organelles bounded by membranes. And such cellular inclusions are the glycogen particles. Glycogen is a polymer of glucose, and the hepatocytes in the liver usually store the glycogen in large quantities, and then they hydrolyze it and release the glucose when it is needed elsewhere in the organism. And the glycogen particles, in wherever they are, they can be visualized with the PAS, periodic acid sheaf stain, that stains the carbohydrates. Then there is the melanin. Melanin, it's a pigment. It's a brown pigment that gives the color of the skin. It is actually produced by a biosynthetic pathway that up to a point it's identical to the pathway of synthesis of dopamine, a neurotransmitter. So, and this melanin, it's first enclosed into the so-called melanosomes, which are membrane-bounded structures, but then it ends up being present in some large structures that are taken up by the keratinocytes in the epidermis. We'll see this in 3 weeks from today, I think, 3 weeks from today. And there it shuttles between the periphery of the cell and the nucleus, because the melanin is the one that protects the DNA in the nucleus from the damaging ultraviolet light. Then there are the fat droplets that are also considered cellular inclusions because they are lipid droplets, but they are not bounded by a real membrane, the trilaminar unit structure. And then there is the lipofuscin, which is simply aggregated denatured protein that accumulates in cells that live long, and cannot be disposed in the lysosome or by the proteasome, which is another structure that may degrade the proteins. And this lipofuscin, it's present in neurons. They live a long life. It's present also in muscle cells. And then the final inclusion that I talk about, it's the hemosiderin. Hemosiderin accumulates in macrophages in the spleen, and those macrophages they phagocytose, they're the effete red blood cells. They are no longer functional, and they digest them in their lysosomes, but they cannot really digest the iron containing core of hemoglobin, and that is the hemosiderin. Now, very fast, I'm going to the cytoskeletal elements that are present in the cells. The cytoskeleton is made of actin filaments, of microtubules, and of intermediate filaments. And the actin filaments are stained here in red, and they form actually bundles. They are play an important role in the motility of cells. They play an important role in generating the shape of the cells. The microtubules, they are stained here in green. They have a polarity. They usually have a minus end that it's at the centrosome, and they extend towards the periphery with the fast growing or plus end. And this polarity is actually recognized by the microtubule motors, and the kinesins, they move on microtubules and transport cargo towards the plus ends of microtubules, and cytoplasmic dynein moves cargo towards the minus end of microtubules. Then there are the intermediate filaments that I drew them here. They sort of surround like a cage the nucleus and extend throughout the cell, and they have various functions, but they also play a role in generating the shape of the cells. Now here I have a video clip that shows the synchronized division of Drosophila embryonic cells in the Drosophila embryo that divide, and that is it's meant to show you the dynamic properties of the microtubules. Microtubules are stained here in green, and you will see that these cells, they divide at the same time. And see, I try to play this movie because it's fun to watch it.
In an early Drosophila embryo, nuclei divide rapidly and in perfect synchrony. In this experiment, both DNA and tubulin are visualized with different fluorescent dyes. After the mitotic spindle has assembled, the microtubules, shown in green, start pulling the blue chromosomes to either pole. The chromosomes decondense and fill the newly formed round nuclei. In preparation for the next round of mitosis, the centrosomes duplicate and migrate to opposite poles of each nucleus, where they form new mitotic spindles, and the process repeats. The whole embryo rhythmically contracts with each division cycle.
Okay, so the blue staining is the DNA that assembles into the chromosomes, and then they separate the sister chromatids, separate the mitosis, and the green are the microtubules. So do you recognize this tune? What is it? It's from Perkin by Grieg, Edward Grieg, Norwegian composer. It fits very well with this dance of the embryonic cells in the Drosophila. Okay, so I think that this was the last slide, and I will stop the recording